Hey guys, today in this video I will be explaining you cell viability and the various assays used to determine cell viability. Cell viability is often defined as the number of healthy cells in a sample. The viability of a cell or population of cell is an overall measure of healthy cells. Alternatively, cell viability can be used to correlate cell behavior to cell number. Cell viability is measured using a variety of techniques and experimental platforms. There are various cell viability assays that are often used for screening collection of compounds to determine if the test compounds have effects on cell proliferation or show direct cytotoxic effects that eventually lead to cell death. Regardless of the type of cell-based assay being used, it is important to know how many cell viables are remaining at the end of the experiment. Viability assays are designed to assess the physical and metabolic state of the cell in order to determine the impact of treatment or culture conditions on cellular homeostasis. The critical link between cellular health and disease underscores the necessity for assays that can measure cell viability in different experimental contexts and model systems. Choosing a cell viability assay from among the many different options available can be a challenging task. Identifying the best cell health assay method to suit your needs require an understanding of what each assay is measuring as a marker, how the measurement correlates with cell viability, and what are the limitations of assay chemistries. Let us understand one by one about these assays. The first one being the ATP cell viability assays. ATP assays are procedures that can measure cell viability based on detection of ATP. All living cells can be detected with ATP assays. Several detection methods can be used, such as colorimetric, fluorescent, and bioluminescent. Most researchers choose bioluminescent ATP assays to measure cell viability due to higher sensitivity, simple and homogeneous protocol, and fast results. These ATP assays are faster than other methods since they do not require long incubation times to convert a substrate into a colored product. They also have excellent sensitivity and broad linearity, making them highly compatible with high throughput applications where low cell numbers are used. ATP is the energy source of all living cells and is involved in various vital biochemical reactions. When the cell die, they stop synthesizing ATP and the existing ATP pool is quickly degraded. Therefore, ATP is widely accepted as a marker of viable cells. Higher ATP concentration indicates higher number of living cells. The reaction step includes luciferin activation with ATP to give a luciferin adenylate and pyrophosphate. This luciferin adenylate reacts with molecular oxygen to yield oxyluciferin in an electronically excited state. The excited state oxyluciferin then returns to the ground state with the release of green luminescent light. The intensity of the luminescent signal is detected using a luminometer. Therefore, higher the luminescent signal, higher will be ATP produced and hence this indicates higher number of living cells. Bioluminescent ATP assays take advantage of the firefly luciferase enzymatic reaction, which uses ATP from viable cells to generate photons of light. Viable cells are lysed to release ATP for detection and reagents containing firefly luciferase enzyme and substrate that are added to catalyze a two-step reaction. Talking about the commercial availability, commercial kits containing reagents to measure ATP are available from several vendors. I have mentioned some of the products and kits with their catalog number as to help finding you guys easy. And for those who are already doing research could be a valuable source. I might have missed some of them, but majorly used ATP cell viability assay kits are shown here. For more detailed information, please refer to the catalog numbers mentioned over here. So let us move on to the next assay method, that is tetrazolium reduction cell viability assay. Over time, the assays for viability have become more complex. Increasingly, dyes which rely on the metabolic activity of cells are gaining favors as they can be performed on adherent cells and therefore lend themselves to high throughput analysis. This assay, that is tetrazolium reduction, uses tetrazolium dyes which can be positively or negatively charged. So it is a colorimetric method which relies on the conversion of tetrazolium into formation crystals. This entire conversion takes place in mitochondria and the incubation time is approximately 4 hours. A variety of tetrazolium compounds have been used to detect viable cells. These are positively charged compounds that readily penetrate the viable cells such as MTT, negatively charged compounds that do not penetrate the cells such as MTS, XTT, WST1. Most of the researchers choose MTT assay to measure cell viability due to higher sensitivity and generates faster results. If we talk about the differences between these two compounds, so positively charged compounds, they readily penetrate the viable cells, whereas negatively charged compounds, they do not penetrate the viable cells. The viable cells, in the case of positively charged compounds, with active metabolism are able to convert tetrazolium dye into insoluble formation product, whereas the negatively charged compounds combines with an intermediate electron coupling reagents such as phenazine methyl sulfate or phenazine ethyl sulfate, which converts tetrazolium to soluble formation product. As the positively charged compounds generates insoluble formation product, so therefore a solubilizing reagent is required to dissolve the formation product, whereas there is no solubilizing reagent required in the case of negatively charged compounds, and I have already mentioned the examples for these assays. So in this assay, the release of formation product determines the number of living cells. So higher is the formation product produced, higher will be the number of cells. So let us look into the principle behind each of these assays. The first and probably best known metabolic dye is MTT, which is developed for a 96 well format that was suitable for a high throughput screening. This assay relies on the conversion of a soluble tetrazolium into insoluble purple formation crystals by reduction. Viable cells with active metabolism convert MTT into a purple-colored formation product in the presence of an enzyme, mitochondrial succinate dehydrogenase, since this takes place in mitochondria. The cellular mechanism of MTT reduction into formation likely involves reaction with NADH or similar reducing molecules that transfers electrons to MTT. When cells die, they lose the ability to convert MTT into formation, thus the color formation serves as an useful and convenient marker of the only viable cells. The next is MTS assay. MTS reagent is used in combination with an intermediate electron acceptor reagent which can penetrate viable cells, become reduced in the cytoplasm or at the cell surface and exit the cells where they can convert tetrazolium to soluble formation product. This happens in the presence of an enzyme mitochondrial succinate dehydrogenase, since this takes place in mitochondria. The cellular mechanism of MTT reduction into formation likely involves reaction with NADH or similar reducing molecules that transfer electrons to MTT. The major advantage of this assay is that the electron acceptor reagents enhances the chemical stability, which allows it to be combined with MTS and form stable solution. Here also the color changes from yellow tetrazolium dye to purple formation product. The next is XTT assay. XTC is also a two reagent assay that consists of XTT reagent and electron coupling reagent. XTT reagent is used in combination with an intermediate acceptor reagents which penetrate the viable cells and convert the tetrazolium into soluble formation product. This happens in the presence of the enzyme mitochondrial succinate dehydrogenase. Since this takes place in mitochondria, the cellular mechanism of XTT reduction into formation likely involves the reaction with NADH or similar reducing molecules that can transfer electrons to XTT. The water-soluble XTT is converted to an orange-colored formation product. This increase directly correlates to the amount of orange formation formed as monitored by the absorbance. The next is the WST1 assay. WST1 reagent is designed to be used for the non-radioactive spectrophotometric quantification of cell proliferation, growth and viability and chemosensitivity in cell populations using 96-well format. So WST1 reagent is also used in combination with an intermediate electron acceptor reagents which penetrates viable cells and can convert tetrazolium to soluble formation product. It has the same mechanism of action as same as MTS and XTT. The WST1 reagent could be reduced to highly water-soluble formation by cellular dehydrogenases in the presence of an intermediate electron acceptor. The mechanism is shown in the figure. The formation product produced by WST1 is dark red color and is more soluble than that of XTT and MTS, which leads to a wider linear range and higher sensitivity. Talking about the commercial kits containing solutions of MTT and solubilizing agents and MTS, XTT, and WST1 and an intermediate electron acceptor reagent are available from several vendors. Those are listed over here. For detailed information, please go through the catalog numbers mentioned over here. You will get all the detailed information regarding the volume, the reaction time, the reaction kit length. Everything will be mentioned in the data sheet. Next is the resazurin reduction cell viability assay. Resazurin is a cell-permeable redox indicator that can be used to monitor viable cell number with protocols similar to those utilizing the tetrazolium compounds. Resazurin can be dissolved in physiological buffers and added directly to the cells in culture in homogeneous format. The viable cells with active metabolism can reduce resazurin into resirufin product, which is pink and fluorescent. Addition of an intermediate electron acceptor is not required in the case of cellular resazurin reduction to occur, but it may accelerate signal generation. The resazurin reduction assay is slightly more sensitive than tetrazolium assays. So care must be taken when measuring fluorescence at higher cell density to avoid the over-reduction of alarmer blue into colorless and non-fluorescent hydroresazurin. If we talk about the reduction of resazurin to resafurin, this is the reaction which takes place, and resazurin as it is blue in color, it gets converted to pink fluorescent residufin. So this pink fluorescence decide the number of living cells. So more is the residufin product, more is the number of living cells. Talking about the commercial kits that contains the solutions of resazurin as well as resazurin powder. These are available from several vendors. So resazurin powder is readily available from the chemical vendors. However, the resazurin dye content and contamination with resirufin can lead to variability in assay results and the need to perform validation of each lot of reagent powder. Viability assay kits containing performance verified resazurin as the primary ingredient are available from different vendors, but the resazurin concentration and additional ingredients vary. So These are the catalog numbers. Please go through the catalog numbers for more information, for more detailed information. Next is the live cell protease cell viability assay. Live cell protease cell viability assay measures the protease activity within the live cells. So measurement of a conserved and constitutive protease activity within live cell has been shown to serve as a marker of cell viability. A cell permeable fluorogenic protease substrate, that is glycine phenylalanine aminofluorocoumarine, that can be abbreviated as GFAFC, has recently been developed to selectively detect protease activity that is restricted to viable cells. Live cell protease activity disappears rapidly after cell death, so it is a useful marker of viable cells. The incubation time for this method is about 0.5 to 1 hour, which is shorter than that of tetrazolium assay, that is 4 hours. Because this method does not lyse cells, it allows for multiplexing with many other assays in the same sample wells, including bioluminescent reporter cell-based assays. One of the advantages of GFAFC is that it is relatively non-toxic to the cells in the culture. So, talking about the principle, the GFAFC substrate can penetrate live cells where cytoplasmic aminopeptidase activity removes the glycine and phenylalanine amino acid to release aminofluorocoumarine, that is AFC, and generate fluorescent signal proportional to the number of viable cells. As soon as the cell dies, this protease activity rapidly disappears, thus making this protease activity a selective marker of the cell population. The non-toxic nature of this GFAFC substrate makes it an ideal candidate for multiplexing with other assay technologies using a sequential assay protocol. So this is the commercially available cell titer floor cell viability assay, which has been available by Promega Corporation. Please go through the catalog number for more information. The last one is the real-time assay. It measures the cell viability in real time. The main advantages of this assay are that it allows simple kinetic monitoring to determine dose response using fever plates and cells, and also because this method does not require cell lysis, the same cells can be used in additional cell-based assays or downstream applications. It utilizes an engineered luciferase derived from marine shrimp and a small molecule pro-substrate. So here I have explained the principle. The pro-substrate is not a substrate for luciferase, but viable cells with active metabolism reduces the pro-substrate into a substrate which diffuses into culture medium where it is used by the luciferase to generate a luminescent signal. This figure here shows an illustration of this assay concept. The reagent is well tolerated by the cell and is stable in complete cell culture medium at 37 degrees Celsius for at least 72 hours, which enables the measurement from the same sample for days without replenishing the pro-substrate. In this assay, an engineered luciferase and a pro-substrate are added directly to the culture medium. The pro-substrate can penetrate cell membranes and enter cells. However, only viable cells with active metabolism can reduce the pro-substrate into a substrate for luciferase. The substrate then exits the cell where it is used by luciferase in the detection reagent to generate a luminescent signal. The same wells can be measured repeatedly for 3 days. The commercial kits containing the pro substrate and the engineered shrimp-derived luciferase are available from Promega Corporation. So, variety, so there are variety of assay technologies available that uses standard plate readers to measure metabolic markers to estimate the number of viable cells in culture. Each cell viability assay has its own set of advantages and disadvantages. The ATP reduction the ATP detection assay is far by the most sensitive, has fewer steps, and is the fast to perform and has the least amount of interference, whereas the tetrazolium or rizazurin reduction assays offer less expensive alternatives that may achieve adequate performance depending on the experimental design. The fluorogenic cell-permeable protease substrate is far less cytotoxic than that of the tetrazolium and rizazurin compounds, while enabling many possibilities for multiplexing other assays to serve as orthogonal and confirmatory methods. Regardless of the assay method chosen, the major factors critical for reproducibility and success include: first, using a tightly controlled and consistent source of cells to set up experiment. Secondly, performing appropriate characterization of reagents, concentration, and incubation time for each experimental model system. I hope this video has made you clear about cell viability and the various assays used to determine cell viability. These are the most preferably used assays till now in this concept. Please do let me know in the comment section. Please do let me know if you have any questions regarding this topic. Thank you.